Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, right, so we're currently in the process of discussing what the actual uses of cholesterol are in the body. Okay, so we've discussed how cholesterol is this essential component of cell membranes all over the body, okay, and therefore since we have a continuous turnover of cells, particularly in the skin and intestine, where we're having to continuously produce new cells because cells are being shed away, okay, we're going to need a dietary input of cholesterol or some other way of producing cholesterol so that we can produce the new cholesterol that is needed in those cell membranes. Okay, right. What I now want to talk about is the other major use of cholesterol within the body, which is the synthesis of bile acids and bile salts from cholesterol that occurs in the liver, basically. Okay, and I want to clear up the confusion between what a bile acid is and what a bile salt is, because they're not the same thing, basically. Okay, right, so this is an activity which can be done by the liver then, so I'll just draw a little picture here of the liver to remind us that this is going to be done by hepatocytes in the liver. Okay, so I'll just label it up as the liver. So hepatocytes in the liver then can convert cholesterol into bile acids, which can then be used to create bile salts. Okay, so there are two main bile acids, which are cholic acid and chenodeoxycholic acid. Okay, and these can be used to make 4 bile salts, which we'll discuss in a moment. Okay, so let me draw a little flow diagram here then. Okay, so hepatocytes are going to use cholesterol, okay, to create firstly what are known as bile acids. Okay, and I want to draw out the structure of the two major bile acids. So the two major bile acids are called cholic acid, okay, and then also chenodeoxycholic acid. Okay, and I can show the structure of cholic acid and chenodeoxycholic acid on the same picture, okay, because there isn't that much difference between cholic acid and chenodeoxycholic acid. Okay, chenodeoxycholic acid just lacks one of the alcohol groups that cholic acid has. Okay, right, so I'm going to now draw the picture of cholic acid for you, okay, and then I'll show you all that you have to change to produce chenodeoxycholic acid. Okay, right, so this is going to be synthesized from cholesterol in hepatocytes, well, both of these are, okay, so they are both sterols, okay, so they have the same core sterol structure, so we'll draw that out first. So here is ring B here, okay, then we've got ring C here, and then finally ring D over here, okay, and then we'll of course have the alcohol group coming off carbon number 3 right down here. Okay, right, so these molecules, cholic acid and chenodeoxycholic acid, the bile acids, they're going to share a lot of the structural features of cholesterol, or rather they're going to have similar structures to cholesterol but modified. Okay, now some of the structures that have not been modified are the two methyl groups that come off carbon number 10 here and carbon number 13 there. Okay, so those remain the same, okay, so we don't change those from cholesterol, they stay the same, but we do get rid of that double bond between carbon 5 and carbon number 6, and we also alter the side chain of carbon number 17 here. Okay, we turn it from that 8 carbon side chain that you had in cholesterol to just a 5 carbon side chain, which is actually going to contain a carboxylic acid group. So let me show this here. So here's carbon number 1, carbon number 2, 3, 4, and here's the 5th carbon, and that's now going to actually be involved in a carboxylic acid group. Okay, so I might just show this like so. So this carbon, at that point there, that 5th carbon, is going to basically be a carboxylic acid group. So that's very different from what we had in cholesterol. Okay, and this is clearly why both of these are considered acid molecules, because they have that carboxylic acid group there. Okay, right, now, that's not finished yet. We need to add 2 more structures on to get cholic acid, only 1 to get chenodeoxycholic acid. Okay, so to get cholic acid, what we have to do is we add an alcohol group onto this carbon down here, which is carbon number 7, 1, 2, 3, 4, 5, 6, 7. Okay, and we're also in cholic acid going to add an alcohol group onto this carbon over here, which is carbon number 12. Okay, so that molecule that you now have sitting in front of you, that's the structure of cholic acid, and it's derived from cholesterol in the hepatocytes. Okay, now to create chenodeoxycholic acid, you only make one modification. Okay, you get rid of this alcohol group coming off carbon number 12 here. Okay, so get rid of that and replace it back again with a hydrogen atom coming off that carbon number 12, and then you've got chenodeoxycholic acid. So those are the two bile acids that are synthesized in the hepatocytes of the liver. Okay, now these can be conjugated with molecules. Okay, specifically they can be conjugated with glycine and taurine to produce bile salts. So let me now discuss this. So basically for both of these molecules, cholic acid and chenodeoxycholic acid, you can conjugate them to the molecule glycine, the amino acid glycine, to produce the glycine conjugates. And the glycine conjugates are then considered bile salts, and they're going to be secreted into the bile and then into the duodenum, of course, to help in emulsification, and we'll discuss that in a moment. Okay, right. So let me discuss how you can create glycine conjugates then of cholic acid and chenodeoxycholic acid. Okay, so firstly let me just discuss the structure then of the amino acid glycine. Okay, so I'll draw a very simple picture here of the amino acid glycine. So glycine is a really simple proteinaceous amino acid. So I'll start by just drawing the core amino acid structure for proteinaceous amino acids. So here's the alpha carbon, here's the amino group, here's the carboxylic acid group, and the alpha carbon also obviously has a hydrogen coming off it. And then the R group in the case of glycine literally just is a hydrogen coming off that alpha carbon there. Okay, so that's why it's the simplest of all the proteinaceous amino acids. Okay, so this is the amino acid glycine here. Okay, and what we now want to do to form glycine conjugates is react this with our cholic acid or our chenodeoxycholic acid. Now, how can we do that? Well, we can react the amino group with the carboxylic acid group of the cholic acid or the chenodeoxycholic acid. Okay, to create an amide link between this molecule and this molecule. Okay, and the two molecules that you can then produce, if you conjugate glycine onto cholic acid, you get a bile salt now called glycocholic acid. Okay, so that's what glycocholic acid is, just cholic acid reacted with a glycine molecule via an amide link between the carboxylic acid group of the cholic acid and the amino group of the glycine molecule. Okay, and glycocholic acid is now considered a bile salt. Okay, so these are the bile acid molecules. These molecules we're now producing are going to be the bile salts. Okay. Now you can also do this to chenodeoxycholic acid, and you then get what's called glycochenodeoxycholic acid. Okay, so the name gets even bigger. So if you conjugate glycine onto chenodeoxycholic acid rather than cholic acid, you get glycochenodeoxycholic acid, which is also another example of a bile salt. Okay, so there's two examples of bile salts. Okay, the other main two examples come from conjugating cholic acid and chenodeoxycholic acid with another molecule. Okay, and the other molecule might be less familiar to you. Okay, which is taurine. Okay, so you can also conjugate taurine onto these cholic acid and chenodeoxycholic acid molecules. Okay, so let me now just discuss the structure of taurine then. So again, taurine has an amino group, so it's quite similar to glycine. It's got an amino group like so. Okay, then it's got two methylene groups rather than just one. So I'll draw this like so. Here's one methylene group, a C with two hydrogens coming off, and then we want two of them, so really an ethylene group there. Okay, and then instead of the carboxylic acid group, you're going to have a sulfate group, basically, in the case of taurine. Okay, so you'll have a sulfur atom double bound to two oxygens, like so. Okay, and then with an alcohol group coming off the end here. Okay, so that molecule that we've got there is taurine. Okay, and again you can link taurine onto the cholic acid and the chenodeoxycholic acid via amide links with this amino group. So it's always this amino group that we're using. Okay, so you can attach this amino group onto cholic acid or chenodeoxycholic acid via an amide link and then create some new bile salts. Okay, so what are these bile salts called? Well, when you conjugate taurine onto cholic acid, you get a molecule called taurocholic acid. Okay, and then of course when you conjugate it onto chenodeoxycholic acid, you get taurochenodeoxycholic acid. So taurochenodeoxy and then cholic acid. Okay, and these are two more examples then of bile salts, this time made by conjugating taurine onto the bile acids rather than glycine. So there are 4 examples of bile salts: glycocholic acid, glycochenodeoxycholic acid, taurocholic acid, and taurochenodeoxycholic acid. Okay, which are all synthesized then by from cholesterol initially and by the liver. Okay, right. So why does the liver synthesize these bile salts? Well, of course, it synthesizes them so it can put them into bile. But what is the purpose of bile? Okay, so let's now discuss what the purpose of bile is, why we need it. Okay, so it's extremely important in the emulsification of fat in the small intestine. So let me explain what I mean by this. So let me just draw a little picture out then of the gastrointestinal system. So here is the esophagus, here is the stomach here, like so. Okay, then we'll have the different sections of the intestine, and I'm going to draw all 3 sections of the small intestine here. We won't need the large intestine, but we will need all 3 sections of the small intestine because I want to discuss the enterohepatic circulation, okay, and that involves the final portion of the small intestine, which is the ileum. So that's why I'm going to draw all 3 different portions of the small intestine out like so. Right, so I'll just colour these in a moment and colour code up the different portions after I've drawn this out. Right, okay, so this is obviously the stomach up here in orange. I will just label it just in case. Okay, so this is the stomach. Okay, then obviously you have the pyloric sphincter, which I won't bother putting on. Okay, and then that leads into the duodenum, which is this first portion of the small intestine, which is in green here. Okay, so this is the duodenum, and that goes right up until this sort of portion here. Okay, so this is the duodenum, the first portion of the small intestine. Okay, then after the duodenum, what you have is a portion called the jejunum, and that goes from the end of the duodenum here all the way on down to here. So all of that is the jejunum, okay, or the jejunum, depending on how you want to pronounce it there. Okay, and then the final portion then of the small intestine after the jejunum is what's called the ileum, and we will need the ileum if we're going to discuss the enterohepatic circulation, which we are going to. Okay, so this is the ileum, this last little portion here, and then of course it will go into the cecum, which will be here, and then you'll have the large intestine after that. Okay, so that's representing the different portions of the small intestine. Okay, so basically bile is going to be released into the duodenum. Okay, so bile will be released into the duodenum, and what function does it actually serve? Well, it functions in the emulsification of fat molecules, basically. So when you consume fat in your diet, what ends up happening is you get these huge fat droplets coming into the intestine, okay? And if you're actually going to break down the fat molecules, which you're going to need to if you're going to absorb them, you need the enzymes to be able to get access to the fat molecules, and it's difficult when they're in these huge droplets, okay? So you want to split the droplet down into many little droplets. Okay, so let me explain what a fat droplet is and why it won't break down into smaller droplets without help. Okay, right, so I've discussed how fat molecules are generally very hydrophobic. They don't mix well with water, and everyone knows this. If you try and mix fat with water, it doesn't work. They separate out into two separate layers. Okay, and this is effectively what happens in the lumen of the intestine. All of the fat molecules coming in your diet aggregate together into these huge droplets, okay? And the outer layer of the droplet is going to contain phospholipids such as lecithin. In fact, one of the major ones that will be there will be lecithin. Okay, so this outer layer of the fat droplet, which I'm now going to zoom in on, so I'm going to zoom in on a little portion of that outer layer there, that will consist of loads of phospholipids like so. Okay, and they'll this time be a phospholipid monolayer rather than a phospholipid bilayer. Okay, so this outer layer of our fat droplet is going to be a phospholipid monolayer. Now, why is that advantageous? Why does it form like this? Well, of course, outside of the fat droplet, you're going to have water molecules. Okay, so that's why you have these phospholipids forming this monolayer on the outside, so that these phospholipids can have their polar heads facing out to where the water molecules are, and then they can have their hydrophobic tails here facing into the core of the fat droplet here, which is where the really hydrophobic fat molecules are going to be. So basically, in this outer layer of the fat droplet, you're going to have these amphipathic lipid molecules, such as phospholipids like lecithin that you've consumed in your diet, okay? Which will be protecting the really hydrophobic lipid molecules, which will be in the core, basically. So you'll have a layer, basically, of polar heads on the outside, and I might even draw this. Okay, here is this layer of polar heads on the outside of this phospholipid monolayer. Okay, and then the hydrophobic tails of the phospholipids will be facing into the core of the fat droplet to interact with the really hydrophobic lipid molecules. So what then are the really hydrophobic fat molecules that we're going to have in this great droplet? Well, the really hydrophobic ones are the triacylglycerols. Okay, so we're going to see a lot of these in this video as well, so we might as well introduce them properly. So triacylglycerols. Okay, which are often abbreviated down to TAGs for short. Basically, these molecules consist of a glycerol molecule, which we've met, bound to 3 long-chain carboxylic acids, like so. So in my cartoons, we show it like so. Here is the glycerol molecule in green, okay, and here are the really long hydrophobic tails, basically, these long chain carboxylic acid molecules that are bound onto the first, second and third alcohol groups of the glycerol molecule. Okay, so this is our triacylglycerol molecule in this core. So the core of the lipid droplet will be full of these triacylglycerols, which are really hydrophobic. They don't really have that much polarity at all. Okay, the entire structure is extremely neutral, basically. Okay, so they're really hydrophobic. They can't interact with water very well at all. So they are stored in the center of the fat droplet here, and then they're protected on the outside by this phospholipid monolayer. The hydrophobic tails of the phospholipids will be facing in to interact with the hydrophobic lipid molecules within, and the phosphate groups will be protecting them from water on the outside. So basically, when you eat a fatty molecule, you're going to be getting these huge, great fat droplets coming into the intestine, basically. And this makes it very difficult to digest down the fat molecules in this fat droplet. Okay, because how are the enzymes supposed to get into where the triacylglycerols are? We need to break down the triacylglycerols into free fatty acids and glycerol molecules in order to get them into the cell. Okay, but it's difficult for the enzymes to get access to the triacylglycerols when they're in these huge great fat droplets. So what we now need to do is break this massive great fat droplet down into loads of tiny little fat droplets like so. Okay, now what's the problem with doing that? Why doesn't the fat droplet just break down into loads of tiny little droplets like so? Well, the problem is that loads of tiny little droplets have a much bigger total surface area than the massive droplet. Okay, so if you add up all the surface area of these tiny little droplets, okay, that would be made from this one massive droplet. That's much, much bigger than the surface area of the massive droplet, basically. Okay, so if you want to split your massive droplet into loads of tiny little droplets, you need to have a lot more of these amphipathic molecules to make the outer shell, the phospholipid monolayer, as it were. Okay, now we don't have enough is the problem. That's why these fat droplets don't break down into these smaller ones, because you don't have enough of the amphipathic molecules. So that's the idea about what bile is, basically. You dump in a huge number of these amphipathic molecules. These bile salts that you're going to dump in, they are going to be amphipathic molecules. Okay, they can also make these monolayers surrounding the lipid core of these lipid droplets, just like phospholipid molecules, basically. Okay, so that's the idea behind what bile is. You dump in this huge number of amphipathic molecules so that these fat droplets can then break down into loads of tiny little fat droplets. You effectively give the molecules which can make the outer shells of these smaller fat droplets. Okay, and that's what the bile salts are for. Okay, right. Now, so every time you take a fatty meal, you will be putting loads of bile salts into the duodenum. Where are these bile salts reabsorbed? Are they just passed straight out of the feces? No, most of them are reabsorbed, okay, and they'll be reabsorbed in the ileum. Okay, so that's why we needed the ileum here. Okay, so reabsorption of bile salts occurs down here in the ileum. Okay, however, not all of the bile salts that you put in will be reabsorbed, basically. Some of them will pass out in the feces, so every time, basically, you secrete bile into the intestine, you are losing bile salts to the feces. Okay, so not all of them will actually be reabsorbed, and that's why you need a constant supply of cholesterol. Well, one of the reasons why you need a constant supply of cholesterol, so that you can remake new bile salts that will replace the ones that you're losing every time you use the bile salts for this emulsification of fats. Okay, now, a piece of key terminology then is the enterohepatic circulation. Okay, so this cycle of bile salts into the intestine for a while, then they'll be reabsorbed back into the bloodstream and they'll go back to the liver. This is the concept of the enterohepatic circulation, basically. Okay, bile salts are said to circulate in the enterohepatic circulation. Okay, the liver will release them into the intestine, they'll move in the intestine for a while, then they'll be reabsorbed, and then they'll go back to the liver, basically, via the portal vein, the hepatic portal vein. Okay, right, and that's called the enterohepatic circulation, but it's not a closed loop, basically. Every time you put in some of these bile salts, you get less coming back. Okay, so the liver is all the time, whenever it uses these bile salts, losing bile salts. Okay, and that's why we need a constant amount of cholesterol being produced or being provided by the diet, so that the liver can use that cholesterol to actually make new bile salts, the bile salts that it lost, basically, from using them. Okay, right, so that's another really important use then for cholesterol in the body in producing these bile salts. Okay, so we'll call it there for this video, and then in the next video we'll have an overview then of how cholesterol is transported around the body. Okay, we'll then discuss lipoproteins in general, specifically we'll dwell on LDL, and then we'll move on to cellular cholesterol homeostasis.